Lecture 7, Presentation 3. The remarkable Jane Goodall. The young woman that Leakey had chosen was, in fact, perfectly suited in temperament and talents for primate field research and had loved chimpanzees ever since childhood. As this image shows, for her first birthday, Jane Goodall's father gave her a chimpanzee doll. In 1962, Jane initiated research at Gombe, Tanzania, and pioneered great ape field research. Well, another blonde hair conducting a little more research with that Goodall tramp. Okay, when Gary Larson published this cartoon, there was a huge hue and cry. Everybody was upset at him for disparaging the fabulous Jane Goodall. Well, Jane Goodall came to his defense and told everybody that is her favorite Gary Larson cartoon. And Gary Larson then donated the cartoon to the Jane Goodall Society, and they use it for selling t-shirts and so forth. So good for Jane and good for Gary. Jane Goodall was the first of 3 women, none initially trained in primatology, whose careers in great ape field research were launched by Louis Leakey. The first of these was Jane Goodall, and she studied the common chimpanzees. She pioneered the methodologies that then were given over to Diane Fossey, who went and trained for a brief period with Jane before she went to study the mountain gorillas. And then later on, we have the rise of Biruta Galdikas with the studies of the orangutans. All 3 women got their start from Louis Leakey. Goodall's research at Gombe initiated the longest continuous research on a single population of apes. As of 2016, the Gombe chimpanzees have been studied for 54 years. By 2018, 56 years. That is over half a century. To a certain degree, Goodall's success came from being the first. She benefited from not being a trained primatologist equipped with specific do and don't do methodologies. She based her research methods on the chimpanzees themselves, and she experimented until she figured out the do's and don'ts for working with great apes. She made mistakes, but willingly assessed the situation, adjusted, and had enormous success as a result. One fact comes through. She had the gift of patience, an absolute necessity in field primatology, and I also have to add, field paleontology. And this is my favorite picture of Jane Goodall with Flint at Gombe in Tanzania. From first-hand experience, Goodall learned that a primatologist must be wary of influencing study animal actions. For the first year or so at Gombe, the chimpanzees avoided Jane, or the males charged her. So she decided to provision them with bananas in order to observe them better. This allowed her to come into contact with wild chimpanzees. However, she soon saw that the extra supply of fruit, a chimpanzee favorite food, heightened group tension. The frequency of agonistic, that is aggressive, behaviors had been altered by her research method and did not reflect the normal pace of behaviors in a natural chimpanzee society. So, Goodall adjusted and began the long process of habituating the chimpanzees to her presence. This is standard practice today. I want to make a note here on this, however. Some primate species cannot be habituated. For example, lowland gorillas are very difficult to habituate. They have been in areas where people have hunted them for meat for so long that they tend to run away from humans. And other species, such as howler monkeys, do not require habituation. They stare right down at and through you. They don't care about a human down on the ground hundreds of feet below. Goodall also broke the unwritten code of researcher detachment. Prior to Goodall, if population individuals were identified, as first utilized by Imanishi, the individuals were given numbers, for example, female 111. Goodall was the first primatologist to name the individuals in her non-human primate study population. Like Imanishi, she was strongly criticized for doing this because many animal behaviorists at that time did not believe that non-humans had individual personalities or even emotions. But this simple act, based on the recognition of individual personalities, was important. Since maternal offspring relationships were easy to identify, Goodall began by naming the adult females. Then she gave each female's offspring a name that started with the same first letter as its mom's name. For example, one of the first adult females Goodall named was Flo. Flo had a distinctive face and personality. She held a high social rank and was an extremely successful parent. In fact, Flo was a great mom. Flo and her offspring represent the F matriline. So in this picture, you've got Flo with her baby Flint, her daughter Fifi, and one of her adult sons, Figgin. Flo was already present in the population when Goodall began her research, so we don't know all of her offspring. Her known offspring include 3 sons, Fabian, Figgin, Flint, and 2 daughters, Fifi and Flame. Over the course of decades of field research, Goodall documented the social histories of the F matriline. Figgin turned out to be a politically clever chimpanzee who managed to hold alpha male leadership status several times. And Fifi, like her mom, was a high-ranking female and successful parent. Her offspring included 4 sons, Faustino, Freud, Frodo, and Ferdinand. We know what Goodall was reading in the field. Who became alpha leaders, and 3 daughters, Fanny, Flirt, and Flossie, who produced more than 15 additional F matriline offspring. Goodall established matrilineal data across the entire population. There was a G group and so on and so forth. And this resulted in several surprising discoveries. By tracking matrilineal relationships, Goodall began to establish generational data, age of reproductive maturity, males versus females, developmental phases, for instance, how long is gestation, how long is infancy, how long is the juvenile stage, lifespans, and so forth. These data are perfect for life history studies. Goodall established the fact that chimp life history stages are intermediate between most other catarrhines and modern humans. So allow me to explain the chart over on the left. And at the lowest level, down here, we have gestation length. And you will see that gestation in a lemur is shorter than it is in an old world monkey macaque. And then you get to the gibbon where it becomes elongate, and then you get to the great apes such as the chimpanzees and humans, and our gestation lengths are about the same length. Infancy, again, increases as we go from the strepsirrhine up to the cercopithecus old world monkey, up to the gibbon ape, and then to the chimpanzee, and then to the modern human. And then the juvenile stages right here, notice how long juvenile stages increase. So we go from a fairly short juvenile stage for the lemur. They get pretty rapidly to reproductive maturity or adulthood. It's much longer in the old world monkey, just a little longer in the gibbon ape, about the same with the chimpanzee, and then look at how long our juvenile stage is in humans. And then we get the other bars. These are for adult life stages. And you can see in the lemur that males in blue, so males over here, and then females in the dark green, have about the same lifespan. They live till they're almost 20 years old in the wild. For the macaques, males and females live until about 30 years old in the wild. For the gibbons, it's just a little bit longer than that. And then for chimpanzees, we see something weird. The males live until they're about 40, and the females, it looks like they're dying off before the males, but they're not. What this blue area above the green suggests is a post-reproductive period for females. So they have the same lifespan as males, but there is a period after they've reproduced when they are still part of the group. And so that's in here. And in human females, we see the same pattern and this post-reproductive phase. So after reproduction is no longer possible. Lifespan is about the same as humans before modern medicine for the wild chimpanzees. And gestation, infancy, and juvenescence are strikingly similar. But notice that chimps share one unique trait in common with humans. Females live past the age of reproduction. So gestation is very similar. Infancy is a little bit longer. Whoops, I'm trying to get rid of stuff here. Infancy is a little longer in humans than in apes, but very close. The juvenile stage is very long in humans. And then we get to this post-reproductive phase, which we only see in chimpanzees and humans. What is up with that? Flo lived past her reproductive age and assisted her daughters with childcare, as you see over here with Flo next to her daughter Fifi, who's got a baby and a youngster with her. Since humans are the only other primates known to have post-reproductive females, this has led to hypotheses concerning the possible adaptive value of grandmothers in human evolutionary history. That is, people who have known child care experience, know where foods are, what has happened in the past in terms of environmental changes, maybe too much rain or cold weather, how to get away from it, how to be safe. Think about the contributions that these grandmothers would have made to humans. This brings up an additional point. Goodall established data on one population of common chimpanzees, that is the chimps of Gombe, Tanzania. This generated interest in the behaviors of other common chimp populations. The results have provided comparative data that indicate behavioral variations associated with habitat differences and, perhaps, cultural differences. In summary, Goodall's field research involved continuous long-term studies on non-human primate adaptations in common chimps, which have provided insights on human evolution. Her brand of primatology is firmly anthropological, and she established the methodologies required for conducting field research on great apes. And what have we gained from this? Now you are ready for Lecture 7, Presentation 4.